In this video, we will understand about the self-life study, how to determine the self-life of a food. Suppose you have produced one product and you have to give the self-life of that food as per the requirement of the Food Safety and Standard Act 2006. So, you need to determine the self life, means best before or the expiry date of that product, when that product will be expired, or means after that day you can't sell that product to the consumers, all these things. So that is a important aspects to determine the self life study. And what is actually self life? Self life is nothing but a period of time within which the acceptability of the consumers with respect to its quality, safety, all things are intact. So the shelf life of product begins from the time the food is prepared. So at the time of preparation only the shelf life is started, and its length is dependent on many factors including the types of ingredients and manufacturing processes, types of packaging, and how the food is stored. That is most important, how the food is stored, and what are the ingredients you have used, whether the ingredient is having more moisture, and what kind of procedures you have utilized, whether you have used very aseptic technology or you are having some standard GMP, GHP, all these things are there. So all these processes, ingredients, and packagings, and all these things are important aspect which determines the shelf life. Shelf life is that within the period that food is safe, quality, and it is accepted by the consumers. Then is the shelf life of food related to food safety? So that also you have to decide before going to do the self life study. You have to understand whether the self life study is happening just because of food safety, or what are the regulations relating to self life, which regulations are present, what are the different regulations present, which uh, is saying what about the self life, that you have to determine what, whether you are going to use by date, best before date, all these things. The use by date, this is used for highly perishable foods. Used by dates is used for highly perishable foods, will present safety risk if consumers will accept that food after that day. A food must not be sold if it is past its use by date. After the passing of use by date, you can't sell that product, or you otherwise you will be executed, okay, or you will be fined, penalized. Best before date is what? When you are going to use best before date, means this is used for foods other than those uh, uh, specified above. It is not illegal to sell food that has reached its best before date, means after the best before date also you can sell the product, but the issue is that after best before date the product's quality is not confirmed, means the product safety is confirmed, product is safe, but the quality is not assured, okay. Then backed on and backed for, all these instructions you can label. What does the date mark look like? So all these conditions are there. Then who is responsible for calculating a self life? Whether your government is saying that the self life study has to be done by one external agency, or the manufacturing, the food business operator itself has to perform the self life study, or the authority will do that self life study, all these things you have to determine. Then who influences the self life? Who? The grower, producers, other suppliers, manufacturers, distributors, retailers, all are influencing. Mainly consumers are also influencing the shelf life study. All are involved in influencing this shelf life study because all are affected by this shelf life of the product. Then factors influencing the shelf life of a product, we know microbial growth and non-microbial spoilers like transportations, any moistures, chemical changes, light induced changes, temperature changes, any physical damage, then any other pest or rodents. These are also the factors who determines the shelf life. Then what is a shelf life study that we have to. So there are two main methods which we can use to analyze the shelf life, either by direct method or by indirect method. In direct method, what we do, we keep the food and do different kinds of test, okay, and the conditions each pre-selected. We have to keep different conditions, like suppose that food is going to be stored in retail, then transportation, then wholesaler, different compartment, different stocks is having different conditions, and one control condition will also be there, and all those conditions will be maintained, and the shelf life study will be carried out, and different testing will be done, and we have to calculate when the the life of the body of the food is ended, or where when the food is started spoiling. That all these things are tested in direct method. And indirect method, this approach uses accelerated storage or predictive macrobiological modeling to determine a shelf life. What is that? A accelerated approach means suppose the temperature for storing that food is 27 degrees centigrade, but what you have done, you have increased that temperature to 40 degrees centigrade and analyze that how long that food is stored without destroying its quality. This is one of the method in accelerated conditions, and based upon some calculations you can analyze that how long the food can be stored, because this indirect method is basically performed for those products which are having long shelf life, those products we, because we cannot wait for 1 year or 18 months for calculating the shelf life, because its shelf life is very high and you can't wait for those many years. So that case, what you have to do, you have to accelerate the storage conditions and macrobiological modeling, then only you can analyze and frame the shelf life study. Then steps involved in calculating shelf life by the direct method. First, the before product on market, that time also you can do. Then identify what may cause the food to spoil or become unsafe. What are the different conditions that will make your food spoiled or become unsafe. Then decide which test is to be done, whether you are going to do sensory evaluations or by checking the different unwanted particles like peroxides, free fatty acids, or all those non desirable particles that is present on the food materials, that you can test. Then plan the shelf life study. What that you have to plan the shelf life study means in which locations, which point, what after the time gaps between which places you can pick up the sample and test, go for testings, all these things you can plan and determine the shelf life. Then only you can determine the shelf life. We will understand it broadly. Once product on market, continue to monitoring shelf life. After determine the shelf life, you dispatch your product to the market, and after dispatching also you keep on monitoring the shelf life of that product. Okay, then step by step determinations of shelf life by the direct methods. So there are a number of decisions that need to be made during the study where it may be necessary to seek expert advice. Sometimes you have to take the advice with the experts. Then identify what may cause the food to spoil or become unsafe. What are the different reasons that your food may be unsafe or spoiled? It may be due to raw material, the quality of the raw material, consistency, level of contamination and storage of raw materials which also affect the product. Then product makeup, which ingredients you use and how they behave when combined. Then water activity, how many molecules of water which are free inside your food material for microbial growth, that constitute water activity, not the moisture present in the food. So that also determine what is the level of water activity, whether it is 0.65 or 0.75. If your water activity is more, then the shelf life will be less. That also determines. Then pH, this is a measure of acidity or alkalinity, you know this, and the pH will influence which microorganisms will survive and grow in a food. For example, fresh mussels have a very short life, whereas mussels in vinegar. So different acidity and alkalinity will determine what kind of microbes will develop, and which microbe is developing is also important. Okay, and if a particular microbe will develop, its rate of reaction is very fast, it can grow very fastly, and then the shelf life will be less. All these things are there. Then oxygen availability, that is also important things. If your oxygens if your food material is exposed to oxygens, it may become rancid, then your food is spoiled. And if aerobic microbes are there, they may also develop. So all these things are determining the shelf life. High standard of hygienes are also important at these, as these packaging methods only limit the growth of the spoilage organism that need oxygens. They do not kill them or limit the growth of organisms that grow in the absence of oxygens. For example, the bacteria that cause slime, botulisms, or listeriosis. So chemical preservatives, which chemical preservatives you are used, because different chemical preservatives have different strength. Then process related spoilage, which process you have utilized, whether you have utilized, suppose pasteurization you are doing, and you have utilized ultra high pasteurizations, ultra high technology, and so and you have utilized HTST, or you have utilized that high temperature short time HTST. So different process, different shelf life level is there. So which technology, which process you have utilized, that is also one important aspect to determine the shelf life study. Then packaging material, what kind of packaging material you are using, because the main point is to control the contamination. If packaging material is very intact, then your contamination level will be less, because this package of the food will go to different place, different temperature, different conditions, different kind of storage conditions. You don't know which place it will go. So that also packaging is important. Then storage conditions, that may also vary. Then step 2, which test to use. You need to select suitable tests for determining the safety and quality of the product. Like suppose you are going to test sensory evaluation. Sensory evaluation can be used, and it is a common methods to use. What is the sensory evaluation is what? Organoleptic test, by the use of your eyes, your nose, your tongue, or your hand. These are all sensory evaluations that you can implement. Then macrobiological test also you can do, like standard plate count, all these tests you can follow, like macrobiological limits for food, macrobiological guidelines criteria. So different conditions you can follow. Then chemical test can detect changes in the product's quality throughout its shelf life. Examples of instrumental chemical test include pH, headspace gas analysis, free fatty acids and total volatile nitrogens. Then some physical tests like travel test, drop test, all these things are involved. Then plan the self life study. What tests need to be carried out? All these steps are what you are planning now. Then how many samples will be tested each time, and how many samples will be needed for the whole study period. All these things are planning. Then when will the study will run, when the product will be produced, then your study will be done, or after how many days you have. These are all planning. Now the study is fully planned and timetabled. Now you have to do what? You have to run the self life study. So during the study, samples should be stored under normal conditions, same conditions as your normal production samples from manufacturing through to consumptions. If this is not possible, the samples should be stored at a non temperatures and humidity. So you have to store that food in normal conditions. These need to be checked and recorded regularly. Regular check also you have to do at the same at the time set out in step 2. So whatever the plan you have done, based upon that you have to conduct the self life study at the normal conditions of storage. Then determine the self life. Then eventually a point is reached when the product no longer meets the quality standards. When you have stored, after some time, that quality criteria will no longer meets the standard. Then using all the informations you have recorded and observed, decide how long the product can be kept and still be of an acceptable quality and safety. And maximum storage times for quality and safety may not be the same. And most important thing, that the shelf life of product should be whichever is the shortest. So during the study you have found that this product is staying this much time, different times you have gathered, but the shortest time period should be given on the level to the consumers, that the shortest shelf life period only you have to give. Like suppose you have tested it for 15 days or 20 days or 1 month, and you have found that after 15 days your product is safe, after 20 days also your product is safe, or is having good shelf life, after 30 days also it is having good shelf life, but you have to put 15 days on your label. Okay, then monitoring the shelf life. After fixing the shelf life on the label, on the packaging standards, then after dispatching the product to the market, in between you have to check the shelf life of the product. Then one more important thing is, if you are changing any kinds of process or any kinds of processing environment, or any changes in the ingredient, then you have to again start shelf life study. It is also critical that the shelf life study is repeated after any changes have been made in the production or the processing environment. Then what are indirect methods? See, in indirect methods attempt to predict the shelf life of a product without running a full length storage trial. We are not going to run the full length of the storage trial. Hence, they can be useful for products with long shelf lives. The two most common indirect methods are accelerated shelf life studies. The trial period is shortened by deliberately increasing the rate of deteriorations. We have to increase the rate of reaction deliberately. Knowingly, we have to increase the temperatures. This is usually done by increasing the storage temperature, because if you increase the storage temperatures, what happens, the rate of reaction is doubled. So based upon that, you can calculate the shelf life. If you increase 10 degrees centigrade of the temperature, then there will be twofold increase of the rate of the reactions. So not twofold, tenfold increase of the rate of reactions. So it is twofold only, twofold increase of the rate of reactions. So what happens, based upon that, you can use the calculations of Arrhenius equations and find out the time when your food will be spoiled. And one more is there is predictive modeling. Predictive modeling are mathematical equations which use information from a database to predict bacterial growth under defined conditions. Predictive models can be used to calculate the shelf life of a food. Information on the changes that occur in the product when it deteriorates, the properties of the product and packaging is required for the calculations. So most predictive models are specific to particular type of organisms. So which type of microorganisms are naturally growing in your product, and you can use the database that shows that these microorganisms can spoil your food products within this period of time, and based upon that predictive modeling, you can calculate your shelf life. So with this, we have understood the shelf life study of the food material, how to conduct shelf life study. Thank you a lot.